Hello everyone, my name is Ankur and welcome to RJPT Simlab simulation. In this video, we will study about the meiosis, mydriasis and the local anesthetic effects of drugs on rabbit's eye. At the end of this video, we will be able to differentiate between the effects of different drugs on the rabbit's eye and their effects, such as meiosis, mydriasis and local anesthetics. So now let us start with the video. At first, let us see the introduction part. The introduction says the intricate functioning of the eye and its responsiveness to various pharmacological agents provide a fascinating avenue for scientific investigation. The above line says that the functioning of the eye and its responsiveness to various pharmacological agents, such as pharmacological drugs, provides a fascinating avenue for scientific investigation. It is a very great opportunity for investigation, and this study develops the dynamic interplay of meiosis, mydriasis, and local anesthetic effects induced by the different drugs on the eyes of rabbits, shedding light on the crucial aspects of ocular pharmacology. So, the study about these investigations it plays the dynamic interplay role of meiosis. At first, let me tell you what is meiosis. Meiosis is the constriction of the pupil of the eye, and mydriasis is the excessive or prolonged dilation of the eye, of the pupil actually. So, meiosis is the constriction of the pupil, and mydriasis is the elongation or expansion of prolonged dilation of the pupil. And then local anesthetics. Local anesthetics or anesthesia, anesthetic drugs are the shows the effects by numbing the specific area of the eye, and it can cause a temporary side effect such as dizziness, headaches, and blurred vision. So it is the numbing of the eye, and which is induced by the different drugs on the rabbit's eye. So now, meiosis, the constriction of the pupil, and mydriasis, its dilation, are essential pharmacological responses that play a vital role in maintaining optimal vision. Understanding the mechanism behind these phenomena, it is not only integral for comprehending normal ocular functioning, but it's also crucial for unraveling the potential therapeutic applications of drugs affecting these processes. So the above paragraph says that meiosis, which means the constriction of pupil, and mydriasis, means the dilation of pupil, these are the essential pharmacological responses that plays a very important role for maintaining the optimal vision of the eye, which means if we have any problem in the vision of the eye, so these are the responses that we can observe in an eye that if it shows the constriction of the pupil, which means it has meiosis. And if it shows dilation of the pupil, we can say that it has mydriasis. So by understanding these mechanisms, we can understand the phenomena of the normal ocular functioning, and it also plays a crucial role in unraveling the potential therapeutic applications of the drugs. So why we can study about the drugs and those disturbed eyes. Furthermore, the investigation extends to the local anesthetic effects of the drug, which hold significant implications for the ocular surgeries and treatments. By scrutinizing the impact of various substances on the sensory nerve transmission within the eye, the study aims to contribute valuable insights to the field of ophthalmic anesthesia. So, we can also investigate and understand the effect of the local anesthetics, which shows the numbness of the eye, which holds significant implications on ocular surgery. So, when we have to perform any ocular surgery or surgery related to eye, we can administer or inject local anesthetics, which will numb the eye portion, and then we can perform surgeries. So, by scrutinizing this impact, various substances of the sensory nerve transmission within the eye, this study aims to contribute valuable insights. So now, the utilization of the rabbits as experimental subjects is justified by their anatomical and physiological similarities to human eyes. Rabbit offers a practical and ethical model for studying ocular pharmacology, providing a bridge between laboratory findings and potential clinical applications. So, rabbit shows similar eye properties in the anatomical structure and physiological similarities. And rabbit also offers a practical and ethical. Rabbit is also a practical and ethical model for studying ocular pharmacology. Through this comprehensive study, we aspire to enhance our understanding of the intricate pharmacological dynamics governing meiosis, mydriasis, and local anesthetic of the rabbit eye. The knowledge gained from this research may pave the way for advancement in the ophthalmic drug development and contribute to the refinement of the ocular treatment, ultimately benefiting human eye health. So, in this experiment, we will study about the meiosis, mydriasis, and anesthetic effects in the rabbit's eye via the use of different pharmacological agents. And this study will help in giving the advancement of the ophthalmic drugs development, and contribute in refinement of the ocular treatments. Now, let us see the equipments required for this experiment. At first, we need the apparatus we require. Apparatuses we required for this experiment are droppers, measuring scale, torch, and cotton wool. Then the subject or animal we can say is our rabbit, which should be of 2 to 5 kilograms. This is the range that should be the average weight of the rabbit. Then the drugs. We require these drugs for the experiment. At first, we require the normal saline. The second, we require the pilocarpine. Pilocarpine is a cholinergic agonist which increases the fluid secretion and treats certain eye conditions. Then we have the amphetamine. Amphetamines dilate the pupils for the retinal eye exams. Then we have the ephedrine. Amphetamine is a sympathomimetic amides, which is used for the stimulant of the central nervous system. And after ephedrine, we have cocaine. Cocaine is used for the eye as an alkaloidal cocaine, which means it is used for an anesthetic for its anesthetic effect, for temporarily insensible to pain. Then we have the physostigmine. Physostigmine is an anticholinergic drug, which is a neurotransmitter, and it increases the cholinergic activity in the central nervous system and the peripheral nervous system. Then after that, we have the xylocaine, atropine, epinephrine, and phenylphine. Now, let us see the principle of this experiment. The principle says the theory. Local action of large number of drugs in an eye can be achieved without systemic effect by application of drugs belonging to the antimicrobial, autonomic, or local anesthetic groups. The eye is supplied with both sympathetic and parasympathetic nerves. Sympathetic nerves are the autonomic nerves that triggers the flight or fight responses. And parasympathetic nerves are the part of also autonomic nervous system that controls the rest and digest function and opposing the fight or flight response of the sympathetic nerves. So the above line says that the local action of these drugs can of large number of drugs can be achieved without systemic effect, so which will be given by local effect by giving the application of the drugs belonging to antimicrobial, autonomic, or local anesthetic groups. And these I can supply both sympathetic and parasympathetic nerves. So ciliary muscles are supplied by parasympathetic nerves. When it contracts, the ciliary body is moved inwards and forward because of the lens and bulges forward, and the eye is accommodated for near vision. The opposite effect is produced by the relaxation of ciliary muscles, resulting in paralysis of accommodation. So the above line says that ciliary muscles, there are ciliary muscles. Ciliary muscles are the hair-like structures that are found in the eye, which are supplied by the parasympathetic nerves. When it contracts, the ciliary body is moved. Ciliary body is moved inwards. It moves inwards, and the ciliary body moves, and the forward moves inwards and forwards. And because of that, lenses bulges forward, and the eye accommodates for the near vision. And so because of the ciliary movement of the ciliary bodies, the lens adjusts its focus. The opposite effect is produced by the relaxation of the ciliary muscles, resulting in paralysis of accommodation. So the opposite effect of ciliary muscles results in paralysis of accommodation. Topically applied drugs can affect the eye by changing conjunctival conjunction, papillary size, or light reflexes, corneal sensitivity, and intraocular pressure. So topically applied drugs can affect the eye by changing conjunctival conjunction, papillary size is the pupil size, and light reflexes and corneal sensitivity. And intraocular pressure is the pressure generated inside the eye fluid or eye. So the pupillary size can be measured by replacing the transparent plastic scale in front of eye as close as possible. Atropine and antimuscarinic agents block the effect of endogenously released acetylcholine on the circular muscles of the iris, and muscle ciliary produces mydriasis and spasm of the accommodation, leading to the cyclopegia, but without producing loss of corneal reflex. Cyclopegia is a temporary condition that paralyzes the eye ciliary muscles, which controls the pupil. So it paralyzes the ciliary muscles. Now, what is eye meiosis? Eye meiosis is the eye refers to having a smaller constricted pupil. So, in meiosis, as we know, the pupils contract. So, this condition also called pupillary meiosis. Pupils are black circles in the middle of the eye. With meiosis, the muscles of iris, the colored part of the eye, tighten and around pupil. These muscles control whether the pupils are large or small. Another name for meiosis is pinpoint pupils. So, in this meiosis, the constriction of the pupils occur, and the pupils and what are pupils? Pupils are the black circles inside the eye, in the middle of the eye. And with meiosis, the muscles of the iris tightens, and which is the colored part around the pupil, and these muscles control whether the pupils are large or small. We can also call it as pinpoint pupils. So typically pupils are bigger and smaller when light enters the eye. When there is a bright light, pupil gets smaller. When there is a dark, pupil gets larger. If you have meiosis, pupil stays small even if the light changes. So it adjusts the amount of light. Pupil adjusts the amount of light that enters the eye. If it's small, if you're in front of the bright light, it will get small, and if you're in the dark room, it will get wider, so that large amount of light can enter the eye, and so we can see that in the dark. But if we have meiosis, so it gets already constricted even if the light changes. So the amount of light will not enter properly inside the eye. So the size of the pupil also changes when you look at the object that is close. Pupils get smaller when focused on something that is closer than the farther away. Meiosis of the pupils can be on one side, unilateral, or both sides, bilateral. These conditions can also be thought of as a mononuclear, monoocular, one eye, and biocular, two eyes. Meiosis, pupillary constriction. It is of two types. Parasympathetic stimulation. It does two parasympathetic stimulation, which muscarinic agonist. The drugs are carbachol, pilocarpine. Cholinesterase inhibitors. Cholinesterase inhibitors are neostigmine and physostigmine. Then we have the sympathetic blockade. We have the alpha 1 antagonist, and for that we have the phentolamine. Now, let us know what are dilated pupils. Dilated pupils, which means mydriasis. As we already know, in mydriasis, pupils are dilated, and when they are black center of the eyes that are larger than normal. The condition may be caused by dilating eye drops from the exam, the side effects from a drug medication, or traumatic injury. This can occur by this, but pupils naturally dilate due to the changes in light and emotional events. But unusual pupil dilation could be the result of a medical condition. So if pupils are dilated, the black center of the eyes pupils are larger than usual. Pupil changes in size to control how much light enters the eye. So the colorful part of the iris controls the shape of the pupil. Similarly, pupils also shrinken when focused on the close object. This is called accommodative response. It accommodates the size of the pupil according to the closer object or near object or far object, or in a bright light or in a lesser light or dark light. Mydriasis pupillary dilation is often required for detailed examination for the retina. Two major groups of the drugs are used to cause pupillary dilation, namely muscarinic antagonists. We have the anticholinergics. For that, we have atropine, tropicamide, and sympathomimetics alpha 1 agonists. For example, we have phenylphine. Short-acting, relatively weak mydriatics such as tropicamide facilitate retinal examination. Cyclopentolate and atropine are preferred for producing cyclopegia, paralysis of the ciliary muscle. It numbs the eye for refraction in younger children. Atropine is also used for the treatment of iridosclerosis, mainly to prevent posterior senescence, when it is often combined with phenylphine. Now, agents that dilate the pupil may abruptly increase the intraocular pressure in closed angle glaucoma by causing obstruction to the outflow tract and are constrained in this condition. Patients should be asked whether they are driving before having their pupils dilated. You should be warned not to drive after until the vision has returned to the normal condition, which means agents that dilates the pupil are not to be administered or taken before driving. Otherwise, their pupils will be dilated and a large amount of light will enter their eye and they will not be able to see anything. A local anesthetic is a medication that causes absence of the all sensations, including pain in a specific body part without loss of consciousness, as opposed to a general anesthetic which eliminates all the sensations in the entire body and causes unconsciousness. Local anesthetics are mostly common used to eliminate pain during after surgery when it is used on specific nerve pathways. Local anesthetic nerve block paralysis, loss of muscle function, and also can be induced. So local anesthetics causes the absence of sensation in a specific part of the body. Loss of consciousness also, but in general anesthetics, loss of consciousness does, and which we administer when we have to perform a surgery, where the whole body gets unconscious. But local anesthetics does not get the whole body unconscious. Only the particular area is uh, causes the absence of sensation, and it is eliminated, and it is used for the elimination of pain during or after the surgery. Oxybuc Oxybuprocaine and tetracaine are widely used in the eye as a topical local anesthetic. Proximetacaine causes less initial stinging and useful in pediatric patients. Tetracaine causes more profound anesthesia and is suitable for minor surgical procedure. Oxybuprocaine, or a combination of the lidocaine or fluorescein, is used for the tonometry. Lidocaine, or with or without adrenaline, is often injected into the eyelids for the minor surgery. Lidocaine is also often injected for surgical procedures on the globe of the eye. So these were the examples of the local aesthetics. Now we can see the observation table of this experiment. Over here we have the list of the drugs that we will use: the normal saline, pilocarpine, amphetamine, ephedrine, cocaine, physostigmine, xylocaine, atropine, epinephrine, and phenylphine. And we have the column which shows the pupil size, the dilation or constriction of the pupil size, the reflex with the light reflex and the corneal reflex. Then we will have the observation over here and then the result. Now let us see the procedure part. The procedure part says place the rabbit number one on the table, hold the rabbit from the tongue and place it in the rabbit holder and heads kept outside from the rabbit holder. Now we have to measure the diameter of the pupil with the help of the scale and record the findings. Now then we have to observe the condition of the conjunctiva and elicit the corneal and light reflexes. Then we have to record the findings. Now we have to put a few 4 to 6 drops of the interval of 2 minutes of saline solution on the left eye and the drug as experimental in the right eye by using the dropper. Respectively, after adding the drops, the medial canthus should be pressed for 10 seconds. So the medial canthus is the corner of the eye where the upper and lower lids meet. So after that, we have to record the following parameters. After 10 minutes, and tabulate the result after inserting the saline and drug respective eye. Diameter of the pupil. The diameter of the pupillary size is measured by a scale, and then light reflex. The effect of the light reflex were examined by holding the torch near to the eye and moving the light beam to and for. And then corneal reflex. The corneal reflex was examined by touching the side of the cornea with a cotton swab or a tip. The rabbit immediately progressively closes the eyelid. Now we have to record your observations in the tabular form. We have to repeat the same procedure for other drugs on separate rabbits. From number 2 to 10. The observation should be taken in dim light and entry of the light rays directly into the eye should be avoided. Now let us see by performing this experiment. And we will perform this in our RJPT Simlab's simulatory laboratory. As we can see here, this is the UI interface of our simulatory laboratory. On the left side of the toolbar, we can see the different rabbits. First is of 2.3 kgs, then 3.1 kg, 4.4 kg, 3.8 kg, and when we will scroll it, we'll have our fifth rabbit, which is of 4.9 kg. On the right side of the toolbar, we can see the rabbit holder, dropper, cotton wick, and torch. And on the bottom side, we can see the instruction part where the procedure instruction will be placed and we will perform this experiment on the basis of that. On the top, we have the observation drop down table. When we will click it, the observation will be over here. We have the factual observation on the rabbit right eye. We can click on the R, then we will see the observation for the right eye of the rabbit. Then when we click L, we will have the observation of the left eye. The factual observations are that these are the theoretical observations. And when we'll click the user input observation right and left, we will see the observation that we have recorded over by performing this experiment over here for the right eye and the left eye. And also we can save the observations by clicking this button. By clicking the procedural flow button, we can see the list of the flowchart of the procedures. So these are the buttons where we can, when we will click it, we can jump to that step, directly jump to that step. So now we will see perform this experiment. On the left side of this, we can see a new rabbit button. So by clicking this, we have a new rabbit. So for each next drug. So now let us perform this experiment. At first, we will place the rabbit in the lab. We will click and the rabbit will be placed over here as we can see here. Now we have to drag the rabbit holder. We will drag it. So rabbit is in the rabbit holder. Now we have to drag a torch. We will place it over here. So now we have to click the torch to grab it with the hand. So now we will grab it and now we will check the light reflex on both the eye. For right eye, we have to click R, and for left eye, we have to click L. First, we will click on the right eye. As we can see here, we will click on the right eye. We will flash the light. And now we have to answer. Light reflects in the right eye. Yes, it has shown the reflex in the right eye. And now on the left eye, the reflex is the closing of the lid of the eye. So when we will flash it, the rabbit will close its eye. So this is a reflex and we have to enter present. The reflex is present. Now we have to drag a cotton wig from the lab. So this is the cotton wig. We will place it over here. Now we will hold it. And similarly, like the torch, we will check the corneal reflex for the both the eyes. On the top, we can see there is a camera button which shows the right and left eyes observation by zooming it. So first we will check for the right eye. As we can see here, it shows for the right eye, the reflex is present, and for the left eye also we will check it. It is also present on the left eye. So now we have to drag the dropper. We will click over here, the dropper is here. Now we will hold the dropper, and now we will select a drug from the list and click on the beaker to get the drug in the dropper. So here is the drop down menu from where we can select our all the different drugs that we have seen before. So we'll start with the xylocaine drug. We'll click xylocaine, and over here we have the normal saline solution. So we'll click in the beaker. It will We will put 2 to 3 drops of xylocaine 1% weight by volume drug in the right eye for 4 times. We have to inject it in the right eye for the 4 times, which is the test eye. We will also put saline 4 times in the left eye. We will put the saline solution in the left eye. So now we will click on the right, and as we can see over here, we will place the xylocaine drug, and we will wait for 2 minutes for 120 seconds. We can also skip this timer. And again, we will select the drug and place it on the right eye. Yeah. We have to perform this for 4 times. Again, we will skip the timer. And now again, we will place it for the third time. As we can see, the eyelid moves. Again, we will skip the timer. And again, we will insert the xylocaine for the fourth time. Now, after doing this, we will skip the timer and we will inject the normal saline solution on the left eye for 4 times. Again, we will skip the timer for 2 minutes, and again, we will put the saline solution on the left eye. So now we can differentiate it and compare it the xylocaine drug with the normal saline solutions effect. We will skip the timer, and again we will collect the saline solution and place it on the left eye of the rabbit, as we can see in the zooming option also. You will skip the timer, and again for the last time we will place the saline solution into the left eye of the rabbit. We will skip the timer, and now we will check the pupil size and click on the by clicking the pupil measure button. So this is the pupil measure button. We will click it. First we will measure for the right eye. As we can see, a scale is here in millimeter, and we have to submit it manually. So as you can see, it lies from the 6 to the 12, so which shows 6 millimeters the size of the pupil. We will submit, and now we will check it for the left eye, which is of the saline solution. It shows the similar, but which is from between the 5 to, or we can say it's from 6 to 11, so which is 5 millimeter, and we will submit it. So now we have checked, and now we will check with the torch. We will check the light reflex using the torch from the right eye first. We have seen that light reflex reflex, and again on the left eye. As we can see, the reflex was also seen over here. We will click the present, and again now we will use the cotton swab. We will hold the cotton swab and we will check first on the right eye. The reaction reflex was shown, and then on the left eye. Again, we will see the reaction of the eye. It blinks, the rabbit blinks on the left eye also, and the reflex is present. So now we have completed the first test on the rabbit one with the xylocaine drug. So now we have seen that it has dilated the pupil a little bit. So now we'll move on to the next rabbit. For that, we will click on the next rabbit button, which is over here. And now we will choose the 3 kg rabbit. Again, we will place it on the rabbit holder. And now we will torch both the eyes before injecting any drug or saline solution. And now we will record the reflex, which is present. Now in the left eye, which is also present. We will click the present. And again with the cotton swab on the right eye at first. And then then we'll click present and then again in the left eye. So as we can see, the reflex has been shown. So now we will check the reflex on both the eye, on the left eye and on the right eye. So here we can see. Now we will click the present button, and now we have checked on both the eyes. Now we will hold the dropper and we will select our drug solution. First we have used xylocaine, now we will use pilocarpine. Now, similarly like the previous, we will inject the pilocarpine 2 to 4 times on the right eye. As we can see on the camera also, we are injecting the pilocarpine, which is of 4% weight by volume. We will skip the timer and again we will inject it on the right eye. Again we will skip the timer and again we will select it for the third time and inject it on the right eye. As we can see on the camera also, again we will skip the timer and inject it for the last time. Insert it into the last time for the rabbit's eye. As we can see here also, the pupil has been constricted. Now we will put the saline solution onto the left eye. So that's how we can compare the effect of a normal saline solution with the pilocarpine solution. We will skip the timer over here. We have to perform it for 4 times. We will skip the timer again. Again, we will place it on the left eye for the third time. Again, we will skip the timer and we will perform it for the fourth time. Again, we will skip the timer. And now we will measure the pupil size with the scale. First, we will measure of the right eye, over which we have placed our pilocarpine drug. So it shows it has shown 3 millimeter size of the pupil. And we will click for the submit button and now we will measure it for the left eye, over which we have placed a normal saline solution. It shows from the 5 to 11, which means 6. So now we'll check the light reflex for the right eye. As we can see here also, it shows the reflex. We will click present and then we will check again for the left eye. On the left eye also it shows reflex. We will click present and now we will hold the cotton swab, and with that also cotton swab, we will check it on the right eye. At first, the corneal reflex, it shows the corneal reflex. We can enter the present and then again for the left eye. Here also we have seen, so we will click present. So now we have checked the difference between the second drug also, pilocarpine. So the pilocarpine is a constricts the pupil. As we can see here, the constricted pupil on the right eye and the normal saline solution shows the normal eye. So now we will move on to the next rabbit by clicking this button. And now we will use the 4.4 kilogram rabbit. Again, we will place it in the holder. Again, we will torch it before administering the drug on the both right and the left eyes. Just like the similar we have performed for two rabbits. It shows the reflex present. Then on the left eye, again it shows the reflex. Now with the help of a cotton wick, we will check for the right eye. We will select the first the cotton swab. Then we will click on the right eye button. So here we will check with the cotton swab. It shows reflex, corneal reflex. We will click present and then again on the left eye, so that we can observe the difference between before inserting the drug and after inserting the drug. It shows the corneal reflex. And now we will place the dropper. We will hold the dropper. And now we will select our drug. We will select our third drug, which is amphetamine. Again, we will take it from the dropper and place it on the eye for the right eye for four times. This was for the first time. We'll skip the timer and again for the second time. Then now after second time for the third time, we'll skip the timer. We'll skip the timer. Now again for the fourth time on the right eye. Again we will skip the timer over there. Now we will put the saline solution into the right eye. Sorry, of the left eye of the rabbit. We will perform this for four times. Again we will skip the timer. We will take the saline into the dropper and we will put into the rabbit's eye. Again we will take the saline solution. We will skip this timer and put it for the third time. As we can see on the camera also. And now again for the fourth time and the last time. Now we will, after this, we will measure the pupil size with the pupil measure button. We will check for the right eye over which we have inserted our drug. As we can see here, the pupil has been enlarged, so which is from 5 to 12. So 5 to 12 is 7, which is 7 millimeter in pupil size of the right eye. Now for the left eye. The left eye has shown no changes, so it is of 5 millimeter size. Now again we will check for the light reflex. First on the right eye, it has shown reflex, corneal reflex, light reflex. And now again on the left eye, as we can see in the camera also, the reflex shows. And again with the, now we will check it with the cotton swab. We will hold the cotton swab and we will check it into the right eye at first for the corneal reflex. As we can see, the corneal reflex has been observed over here. We will click the present button, and on the left side also, we will click, we will see over there the corneal reflex. We will click present, and now we have completed for the test with the third rabbit also. So, by observing the observation table here, we can see for the factual observation of the rabbit's right eye. So, when we inserted the xylocaine on the right eye, we have seen that light reflex, and we have seen there is an absent of the, and after that we have seen the absent of the corneal reflex. We have to observe, and the pupil size should be of 6 millimeter, and result will be of local anesthetic. But when we have performed this, we have observed that xylocaine has shown light reflex and also shown corneal reflex. Pupil size is 6 as the theory, but result has shown no effects. Similarly, for the pilocarpine, it has shown both light and corneal reflexes. Pupil size is 3, should be of 3, and Miosis effect. But what we have observed that pilocarpine has shown both the corneal light and reflex, pupil size is 3 also, and has shown Miosis effect. Similarly, similarly, when we perform for the amphetamine has shown both light and should show both right and left, light and corneal reflexes. Pupil should be of 8 and it should show Mydriasis. But similarly, when we performed it, it has shown the light and corneal reflex, and it has pupil size of 7 millimeter, and it has shown Mydriasis effect. So this is how we can observe for all the drugs. Now again we will perform for our next drug, and for that again we will have our next rabbit, and for that, before that, we are using the torch. Before administering the drug or saline solution, we will check the light reflex on the left and the right eye. At first, on the right eye of the rabbit, it shows reflex. We will click present, and then on the left eye, we can see the reflex. We will enter present. After that, we will place the cotton swab and we will hold it the cotton swab. And after that, we will check the reflex for the right eye. For the corneal reflex, we will observe the corneal reflex over here. We will click the present button. And now for the left eye, we will poke it with the cotton swab. We have seen the corneal reflex and we will enter present. Now with the help, we will hold the dropper and now we will enter our next drug. So for that, we are using cocaine. Cocaine is a local anesthetic. So we will take the 2-3 drops of cocaine and we will administer into the right eye. We will skip the timer and again we will perform for the right eye. We will administer cocaine into the right eye. We will skip the timer and again for the right eye we will administer cocaine. The third drop, and now again for the last, the fourth drop. We will skip the timer and we will administer the fourth drop. As we can see, we have administered all the four drops of cocaine. We will skip the timer and now we will administer this normal saline solution to the left eye so that we can compare the effect of the cocaine with the normal saline solution and we can observe the difference of the effects. Again, we will administer the second drop to the left eye. Now, again, after that, for the third time, we will We will skip this timer. We will administer the normal saline solution to the left eye, as we can see in the camera also. Again, for the last time, we will administer the normal saline solution to the left eye. And now, after administering all the drug solution and the saline solution on the right and left eye, we will observe the observe the pupil. We will measure the pupil. First, we will use first we will measure the right eye. So here we can see it's from the fifth to 12th. Fifth to 12 is 7 millimeter. We will submit it. And then now for the left eye, with which is of the saline solution, it has shown from 5th to 11th, which is 6. We will submit 6 millimeters. Now again, we will check it for the light with the help of the torch on the right eye at first, as we can see in the camera also. For the right eye, we have seen the light reflex. And then on the left eye, here also we can see when we flash the light, the reflex has been observed. We will click on the present. We can also close the camera button here. Now we will use with the help of cotton swab. We will hold the cotton and check the corneal reflex. First on the right eye. As we can see, there is no reflex. So we will click on the absent. And now on the left eye. As we can see, the corneal reflex is present on the left eye. We will click the present. And now we have completed for our fourth rabbit also. Now we will see the observation table here. And now when we perform for the factual observation, when we perform for cocaine, the light reflex should be seen, but the corneal reflex should not be seen. Pupil size should be of 8, and Mydriasis effect should be the result. And when we have performed it, we have what we have seen. We have seen the light reflex, but we have not seen the corneal reflex, similar to the theory. But we have seen the pupil size of 7 millimeter, and the results we have seen is Mydriasis effect. So it is similar to the factual observation. Then similar for the left eye also. What should we get? The light reflex and corneal reflex of saline solution should be same. Pupil size should be of 6 and result should be of no effect. And what we have observed the same, but the pupil size is just slight different. So slight different has shown Miosis effect. So this is how we understand the difference of the effects of different drugs such as xylocaine, pilocarpine, amphetamine, and cocaine. The drugs which shows Miosis, Mydriasis, and local anesthetics. So these are the drugs through which we can understand the pupil size, the change in the pupil size, or the local anesthetic effect on the rabbit's eye. Why we are performing it on the rabbit's eye? Because it's similar to the human's eye. So this is how we can understand the difference between the effect of different drugs. So now we have come to the end of this video. And you can also understand for more drugs also. When we click on the new rabbit button, when we will click on the next rabbit, we can perform it for the next rabbit also. And we can understand its effect for different drugs too. So now we have come to the end of this experiment. Thank you guys for watching this video. And I hope that you have learned a lot in this experiment about the different physiological agents and its effects on the rabbit's eye, which shows Mydriasis, Miosis, and about local anesthetics too. So guys, thank you guys for watching this video. And for more such learning content, subscribe to our YouTube channel, RJPT Informatics. And you can also follow us on our LinkedIn profile, on Instagram, as well as in Facebook. We update and upload our such learning content over these platforms. Thank you.